Hello everyone, my name is Rajendra Kumari and I am the Global Head of Scientific Communications at Crown Bioscience, a preclinical contract research organisation providing high quality models, technologies and support for your drug development needs. Today's webinar is part of our educational tech talk series focused on in vivo solutions for oncology and immuno-oncology drug development. So there's no Q&A session at the end, but please feel free to ask questions in the chat box and we will answer them as quickly as possible. In my talk, titled Xenografts and Homografts, All Models Are Not Equal, I will discuss the challenges in modelling tumour heterogeneity and the tumour microenvironment, and provide an overview of today's animal models for oncology research and their strengths and limitations to allow the right models to be selected appropriately. The biggest challenge when trying to model human cancer is that it's not a single disease and not all tumours are created equally. Each patient's tumour is unique and with each type being highly complex from different histopathologies and highly heterogeneous, which drives cancer progression and resistance to treatment. Cancer is a genetic disease and each tumour has a mixture of genomic aberrations, which are oncogenic or driver mutations, as well as bystander mutations, which vary from cell to cell, patient to patient and cancer to cancer. There are some actionable aberrations which may be predictive of drug response or resistance and therefore useful for targeted therapy, which is shown by the example here for non-small cell lung cancer. Most mutations are believed to occur early and with clonal selection, there changes at different stages of the disease, for example, after treatment, relapse or metastasis, or further mutations may arise depending on the environmental stresses. Reoccurring tumours, this is following treatment, generally are more aggressive, refractory and lethal, which may also be due to the cancer stem cell population or plasticity of the tumours, resulting in resistance. Somatic mutation frequencies observed in the exomes in tumour normal pairs varies across cancer types, as shown here, as well as within the cancer types, resulting in unique neoantigens. However, tumours can evade immune surveillance despite these neoantigens via immune checkpoints. Cancers like melanoma and non-small cell lung cancer, which have high somatic variations induced by carcinogens such as tobacco smoke and UV light, benefit from checkpoint inhibitors, and these are on the right-hand side of this graph. And this is because of that high mutational load, and as well as other cancer types which have high DNA mismatch repair or microsatellite instability, making them more visible to the immune system once those breaks are released. So the genetic variations in the tumour microenvironment are integral to each other. So the reality is that we're not just treating the heterogeneity of cancer cells, but the tumour microenvironment as well, which needs to be understood and considered when modelling cancer. The diagram nicely illustrates the different components of the tumour microenvironment involving immune cells, endothelial cells, fibroblasts, and mesenchymal stem cells. And the tumour microenvironment also offers potential new or novel targets to drug alongside the targets expressed on the tumours. With our evolving understanding of tumour heterogeneity, tumour microenvironment and plasticity, can our preclinical models, specifically our animal models, embrace this diversity and reflect the complex nature of the disease and those novel targets? So let's dive into what animal models for oncology research are available. So firstly, it can be quite difficult to know which models can be used and when. And historically, models were simple, like murine-based in vivo models or in vitro 2D cell lines, but these were seen as poorly predictive. Then over the last few decades, new models emerged to overcome these challenges, especially the patient-derived xenograft systems, due to improved engraftment of the human tissue in NOD-SCID or NSG mice. And this is also alongside your standard cell line models from those 2D cultures. So the CDX. There are different types of immune deficient mice that can be used for different xenograft modelling, and this all depends on the tumour model. So for more fastidious tumours and tumours fresh from the clinic, more immunodeficiency is required to allow engraftment. And in some cases, you need further supplementation with hormones, growth factors, or basement membrane supplementation. With the emergence of immuno-oncology and new immunotherapeutics, the immune deficiency nature of the xenografts limits their application for immunotherapeutic evaluation, especially for T-cell modulation. So therefore, the demand for immuno-oncology relevant models has led to increased use of different immune-competent mouse models, which are shown at the top, or hybrid chimeric mouse-human systems. Xenografts can be used to evaluate certain cellular therapeutics, such as cellular therapies, ADCs, depending on obviously the mouse strain and the innate immunity, or they can be used in humanized systems where engraftment of the human immune cells on these immune deficient backgrounds enables reconstitution of the human immune system. But there are some challenges and limitations when doing this, and this will be discussed further in the next tech talk. For the purpose of today's presentation, I will be focusing on the ones highlighted in the red boxes. Further complex models at the orthotopic and metastatic sites will be overviewed in the last talk of this series. And you can find more information about any of these models via our databases that are listed here. So let's look closer at these xenograft systems in immune-compromised or deficient systems. And this will be predominantly focused on subcutaneous modeling, which is typically used because it's easy to expand the tumor material and monitor the tumor growth. So the clinical relevance for mouse models varies depending on the model that is used. And for human xenografts, this is dependent on how they were created. So 2D cell lines and their associated cell line models, xenograft systems, have been the cornerstone of drug development, and cells are grown or cultured in 2D to generate these CDXs. And these cells are available from cell banks. They're widely used for research as they're easy to grow, manipulate, and there's extensive sort of track record in the literature. In comparison, PDX are derived directly from patients and must be maintained by serial passage in immune-deficient mice due to the sort of poor adaptation to plastic. And some of these are relatively low passage and new, so PDX characterization can be limited. However, because of their temporal proximity to the patient and having never been subjected to the selection pressures of cell culture, those PDX models are believed to be to have higher fidelity to the patient than CDX models, maintaining tumor heterogeneity and better prediction of treatment outcome, resistance, and disease progression. So the publication on the left hand highlights how the majority of cancer cell lines used in vitro, so for example, the NCI60 panel, lacked heterogeneity, and that all the cell lines either grown in vitro or in vivo looked more like each other than the tissue of origin. And so thus the NCI60 collection was retired in favor of more clinically relevant PDX models. And PDX models, when compared back to the original patient material, showed a very high level of correlation of the gene expression profiles of the patient and the PDX material, as shown here for colorectal cancer. And our bioinformatics team looked at the RNA-seq data from the Cancer Genome Atlas across 7 cancer types, so there's hundreds of data sets here, and matched it against the PDX collection that we have for the same cancers, and it showed high correlation, confirming that tumors hadn't drifted like the CDXs. And in addition, there is a lot of evidence generated to demonstrate the clinical relevance of PDX in the form of co-clinical and retrospective studies where clinical responses could be mirrored by the PDX and with the sort of predictive power of more than 90%. So there are many PDX collections that have been developed to represent patient diversity and heterogeneity, and this includes our PDX biobank called HuPrime for solid tumors and Leukemia for blood cancers. So we have over 3,000 PDX models. There's more than 30 different cancer types, and our goal is to represent diverse cancer types, subtypes, treatment responses, and population and clinical spread. And these tumors are fully annotated for the clinical information, pathology, and treatment history and response, and also genomics. And this can be found on our database called HuBase. And you can search through the gene expression levels, profiles, cancer types, mutations, and treatment. And all of our models are QC checked and authenticated by deep sequencing. There are some unique models with specific mutations, resistant profiles, clinical history, and obviously the publications associated with these are listed. But what also is significant of our PDX collection is the matched in vitro capabilities with organoids for in vitro screening derived from these PDX models or the primary cells, as well as 3D spheroid cultures. And we list the models which are ready for use with their growth curves to help select the right models. And this type of information helps support the model selection for studies such as mouse clinical trials, which will be discussed further in one of our tech talks in the series. And then through our bioinformatics team, you can sort through the responder, non-responder profiles and look at predictive biomarkers. And CDX models are still extremely valuable for early drug development, for providing insights into drug sensitivity, cell biology, and also the signaling pathways. And so we have over 250 well-characterized and validated xenografts models, and this includes subcutaneous, orthotopic, and systemic models, which can be found in our database called Xenobase. Now let's look at the immune competent systems, starting with syngeneics. And these are cell lines as well. So they're cultured in plastic and are routinely used with a vast historical data found in the literature. But unlike human cell lines, there's not that many. So we have a panel of over 30 syngeneic models, nearly covering 20 cancer types, which have been characterized with full immune cell profiling via FACS, RNA sequencing data, proteomics, and checkpoint inhibitor benchmarking. And this is accessible via our MU-based database online. Now, you can evaluate a range of different immunotherapeutics, including checkpoint inhibitors, vaccines, oncolytic viruses, microbiome modulators, so we have germ-free capabilities, cellular therapies, and other immune modulators. And you can do this in combination as well. But there are some limitations. This is murine biology that is not entirely predictable of clinical response, and you can only test mouse surrogates or those cross-reactive agents. And these models have been generated, they're mostly carcinogen-derived, so they do lack relevant oncogenic drivers and the disease progression. But despite these limitations, the syngeneics are still the only viable model system which can be easily set up in immune competent hosts and allow efficacy and mechanism of action studies. And because of this efficiency, we've also developed a higher throughput MuScreen platform, which is run on a quarterly basis. So you run a panel of pre-selected syngeneics. It's cost effective and time efficient and allows you to include multiple arms and multiple models. And the cell lines are easily engineered as well to express human targets or improve immunogenicity. So especially for those colder tumors, generate reporter cell lines to quantify cytokine production, for example, and as well as making them bioluminescent for optical imaging so that you can implant them at orthotopic sites. And this enables the expansion and development of new syngeneic tools for different applications. So most checkpoint inhibitors were profiled in syngeneic models. So for example, anti-PD-1 proof of concept was conducted in MC38. We have a panel of 12 models which have been extensively profiled genomically, immunologically, and phenotypically, all at the baseline and post-treatment using different agents for benchmarking with different checkpoint inhibitors. And this facilitates the easy selection of suitable models. And as you can see, not all syngeneic lines are equal. And we have reported on different intrinsic tumor immunity and mechanism of action for anti-PD-1 through immune cell depletion studies, which is reported in this publication on the right. These models don't necessarily represent the tumor heterogeneity, but do reflect immune diversity and changes in the tumor microenvironment as the tumor grows and responds to the treatment. As mentioned, one of the limitations of syngeneic models is the limited number of models available and the lack of relevant driver mutations. Genetically engineered mouse models, which have high correlation to the clinical disease, are slow, spontaneous tumor models, which can be time-consuming and efficient for pharmacological or efficacy studies, especially large studies, but they do carry relevant tumors and mutations. The MuPrime platform was therefore created to take advantage of this huge collection of gem models to streamline their use. And here, illustrated in the middle, is you can see that this is very similar to the generation of PDX in that the tumors that arise in the site of origin in the gems are harvested and implanted into recipient mice of the same background. Once established, they're grown and then expanded and used for studies in similar controlled manner as syngeneics, but do not adapt to grow in plastic and are maintained in serial passage in vivo. This preserves the original gem tumor histopathology, as shown here for the KPC pancreatic mouse model. So looking at the homograft model in comparison to the original patient at the subcutaneous and orthotopic site, you can see that the architecture of the tumor is retained, and also this particular model carries a P53 and KRAS mutation. Similar to syngeneics, there is model characterization that can be found in MuBase, and also the well-characterized models are included in the MuScreen platform, which is listed in the table. When only human-specific immunotherapeutics are available, or when no mouse is available, then engineered models with human target knock-in are available. At Crown, we have HuGen models, which are immune-competent chimeric mouse models, engineered to express humanized drug targets such as genes encoding for example, immune checkpoint proteins. In some cases, this may be a single knock-in like PD-1, CTL4, LAG3, OX40, etc., or also double knock-ins like PD-1 with CTL4 or LAG3, and this can be used for bispecifics. These can be used to interrogate target engagement, pharmacodynamics, and also efficacy with syngeneic tumors. And we also have HU cells, which are mouse cell lines engineered to express humanized targets, such as PDL1, to achieve fully humanized ligand receptor complex. And details of these models can be found in MuBase, with details of the homozygous, heterozygous knock-in, the human target expression binding to the human antibodies, and also response to therapeutics. So in summary, all cancers are not equal, and neither are the in vivo models of cancer. Cancer is too complex to model, but there are a range of different preclinical tools and models available, both immune deficient and immune competent systems, providing diverse applications and solutions. There is limitations in replicating clinical relevance, with PDX being the gold standard, whereas homografts, such as syngeneics or our MuPrime platform, are tools for immune oncology. So selecting the right model is dependent on their specific features and their applications. So finally, I will leave with this table, which summarizes some of the different features discussed today. So this is shown at the top, and for each model alongside each other, you can see what the highlights are in terms of strengths and weaknesses, as well as the application on the right. So thank you for participating in today's Tech Talk. I hope you've enjoyed it and will join us for the next talk in the series, which is Humanized Mouse Models for Immune Oncology Drug Development, which will be presented by my colleague, Dr. Gerald Fewer. To learn more about our relevant resources, visit the resource section of the video platform and visit us at crownbio.com.